Hi, thank you very much for the opportunity to tell you about CRISPR-Cas systems and the ways that these systems can be harnessed to develop technologies for improving human health. One of the really exciting advances in biology has been the completion of the human genome, and now DNA sequencing technology has advanced significantly so that we can sequence the genome of many individuals. By comparing the genomes of individuals who are healthy with groups of individuals who are affected by specific diseases, scientists have been able to identify many different genetic mutations that contribute to different disease. For example, it's now known that there are different mutations that contribute to sickle cell disease, or ones that lead to cystic fibrosis or different forms of eye degeneration. In fact, nowadays, there are more than 6,000 genetic diseases where we now have identified causative genetic differences. But not all genetic differences are necessarily harmful. In fact, some have been found to protect us from certain diseases. There's a mutation in a gene called CCR5 that is known to protect those individuals who have that genetic mutation to prevent the infection of HIV virus. There are also other mutations that have been identified that are known to reduce the risk for diseases like Alzheimer's or diabetes. As our understanding of the genetic contribution to disease advances, a tantalizing idea is, can we go into the genome of individuals and either remove disease-causing mutations or put in genetic differences that can protect from certain diseases? So, to think about how to do this, let's look at what the human genome looks like first. The human genome consists of many different chromosomes, and this is an artist's rendering of what a chromosome looks like. If you zoom into the chromosome, you'll see that each chromosome is a long string of DNA, and that DNA is composed of just 4 different letters, A, T, G, and C. These are the different DNA bases that make up of the human genome. As scientists start to study the genome, nowadays we can annotate the genes that are on the genome. And so you can think of each long string of DNA that forms a chromosome to be made up of different segments of genes. And each gene has a structure. And within that structure, there are what's called promoters, enhancers, the genes, and the terminator. An analogy for what a gene structure is a sentence. So a promoter is similar to a verb, an enhancer is like an adjective, a gene is like the noun of a sentence, and then the terminator is the punctuation mark. And so each chromosome is a long string of many different gene structures, and it's analogous to a book where you have many different sentences. Each chromosome can be thought of as a chapter of a book. So if we take this analogy further, a genetic difference or a mutation that's found in a specific spot in the genome is like a typo that's found in a specific section of a book. So if one were to correct a typo from a book, then this can be done quite simply using a word processor, where you simply open up the search and replace function. You can type in the typo, and then the search and replace function will place the cursor right at where that typo is. So now you can either delete the typo, or you can type in the new replacement word. So what is the search and replace function for the genome, for a chromosome? It turned out that there are many technologies that have been developed to make gene editing or genome editing possible. And some of those technologies have been developed to correct specific mutations like zinc finger nucleases or tail nucleases. But the latest technology, the one that's the easiest to use, comes from a system called CRISPR. And if some of you have had yogurt for breakfast this morning, you have already encountered CRISPR. In fact, the bacteria that people use to make yogurt carry CRISPR systems, and the producers of yogurt use the CRISPR systems to immunize those bacteria from certain bacterial viruses so that they can become more robust culturing systems for producing yogurt. So here is an animation that describes how the CRISPR system works. Here is a bacterial virus. It latched onto a bacterial cell and it injected its own genetic information into that cell. Inside a bacterial cell, there are these RNA molecules that are called guide RNAs, and they work with a protein called Cas9 to form this immune complex to then search along the invading virus's DNA. If the RNA, which is in red, matches the virus's DNA to form this duplex helix structure, the enzyme will get activated and will make a cut in the virus's DNA. And that destroys the virus and circumvents replication and subsequent spread of that virus. It's this same mechanism that we can co-opt to develop a gene editing machinery. So we engineer the Cas9 protein and we engineer the RNA guide so that this whole system can work and be introduced into the nucleus of a human cell. Inside of a human cell, this RNA guide has been programmed to recognize a specific spot in the genome. And if that RNA finds it and forms this duplex structure, then the Cas9 molecule will make a double-stranded cut in that spot in the human genome. That double-stranded break is the equivalent of a cursor in Microsoft Word. So where that break happens, the DNA can get edited. One way to edit is simply removing a little bit of sequence from the two broken ends and re-gluing it back together. But that small change can be very useful for inactivating a gene that you don't want to have in the cell. A second method is more powerful. So instead of simply gluing the two ends together, you can introduce into the cell a piece of synthetic DNA, like this purple strand here. This strand will join the two broken ends and then allow you to insert a new sequence that replaces the error that we would want to fix. And so these are the two different ways that gene editing happens inside of cells. These methods can be applied through two different strategies to develop therapeutic treatments. One of them is called ex vivo gene editing. The way that this works is you can remove cells from a patient, for example, blood cells, and you can fix those cells and then put them back into the patient. This methodology has been now applied to the treatment of sickle cell disease and also different forms of cancer. And the sickle cell application in patients in the clinical trial environment has already shown that there is quite a bit of promise and is showing efficacy in sickle cell patients. The second strategy is to treat disease in vivo, because some organs, like the brain, for example, or the whole liver, cannot be removed from the patient and then fixed ex vivo and then put back. To carry out in vivo editing, one would deliver the gene editing machinery directly into the patient so that correction can take place directly in those affected tissues. And so there are clinical trials underway as well in both the liver and also in the eye, where this drug developers are trying to treat diseases in the body. So we have tried to see how well these edits can work, and we began by using a mouse model. And we targeted a gene called PCSK9, which is a very prominent drug target that people use to be able to treat high cholesterol and reduce heart disease risk. And there are antibodies that have been developed to deplete this protein called PCSK9. So we thought, instead of depleting the protein by using antibody, maybe we can edit the PCSK9 gene to remove it, to mimic what is naturally found in some people who naturally don't have the PCSK9 protein. So we developed a viral vector. This is an adeno-associated virus. And in the virus, we package in the Cas9 system as well as the guide RNA that's been designed to target the PCSK9 protein. By packaging everything into a virus, we can inject this viral vector into a mouse so that they can target cells in the liver of that mouse. And so this is what we were able to observe. So this is the PCSK9 level in the mouse. This is 13 days before injection, all the way to injection day, which is 0, and then following the progress for 28 days after injection. And if you look at the PCSK9 level in the blood for a mouse that didn't receive the viral vector, you can see that it fluctuates a little bit, but you can continuously detect it. But if you inject in this virus that carries the Cas9 system to get rid of PCSK9, right after injection, 7 days later, you can see that the PCSK9 protein level becomes undetectable. And so this persists for the rest of the time that we were able to measure in the mouse. And so this shows that it's a single injection, and it gives you a very long-lasting effect. A reduction in the PCSK9 protein level should result in a reduction in the cholesterol level. And so this is what the cholesterol data looks like. Here is a mouse that only got the vehicle injection, so it didn't get the Cas9 PCSK9 targeting viral vector. In comparison, for a mouse that received the PCSK9 targeting vector, we can see that cholesterol level dropped by 50%. And so this is exactly what we expected to see because a depletion PCSK9 should result in a very significant reduction in cholesterol. And this should also translate into a reduction in the heart disease risk. And so this represents a one-shot permanent reduction of cholesterol, and this can be used to treat cardiovascular disease. And so nowadays there is a clinical trial underway that has already shown very promising data in non-human primates for reducing cholesterol, and then they're going moving toward going to human patients to be able to test this. So this is just one example, but there are really many other examples or many other diseases where this gene editing approach can provide a therapeutic strategy. And here is just a short list of some of the genetic diseases that we may one day be able to use the gene editing system to be able to treat. So what I told you about so far is this molecule Cas9 and how we're able to engineer Cas9 into a gene editing technology for treating diseases. But CRISPR systems are much more diverse than just Cas9. In fact, there are many different types of CRISPR molecules. Cas9 is found in some bacteria, but if you look into other bacterial cells, you will find other enzymes such as Cas12 or Cas13. Cas12 has also been developed for editing DNA, but both Cas12 and also Cas13 have also been developed as a diagnostics technology for detecting infections from viruses or bacterial pathogens. And these diagnostics technologies, such as Sherlock, provide a cost-effective and also rapid way to be able to read out virus or bacterial pathogen infection. And so this picture just shows different paper strips that are a paper strip test that are being developed to be able to rapidly detect, for example, coronavirus SARS-CoV-2. And so these give you a few of the different examples of how CRISPR technologies have been developed to both advance treatment but also the diagnosis of disease. Of course, Cas9 and Cas12 and Cas13 are just a very small sliver of the diversity that are found in nature. And so one thing that we are super excited about is to look into the natural diversity, the plants, the microbes, the animals that have evolved over millions of years, and identifying novel molecular mechanisms that are found in these biodiversity to turn them into additional biotechnologies that have the ability to advance human health. So last but not least, I just want to take a moment to acknowledge my team. So on the left in the picture are the students and the postdoctoral trainees and also staff scientists who are in my lab, who have been working with me to develop these technologies. And then none of this work would have been possible if it were not for the funding from the Howard Hughes Medical Institute, from Lisa Yang and Hak Tan, the National Institute of Health, Patricia and Jim Poitras, and also Open Philanthropy, and they have generously supported our research over the past number of years. Thank you very much, and I'm really happy that I'm able to tell you about the CRISPR technologies. Thank you.